So let's start in time. Dear friends and colleagues, on behalf of my co-chair, Professor Sandrine Dubrac, I'm very happy to open and welcome you to another of the very successful ESDR Kitchen Live webinar series. And today we have one of my favorite kinds of ESDR kitchen, which is the freshly baked. And you are all aware that this is highlighting rocket science and quite recent publications. And today we selected two rising stars in the field, two very nice papers which came out last year. And we will start with the paper published in Immunity in February 2023 with the first author, Anna Redl. Anna Redl is clinician scientist in the lab of Georg Stary in Vienna, and her special interest is granulomatous diseases. She has the model disease sarcoidosis and published a really very nice publication, which is that nice, so nice that I don't want to take a lot more time from her, but really ask her to start the presentation. After I say thank you very much to UCB, to our sponsor for today, for financing and helping to perform this kind of webinar series. After that, Sandrine will introduce Marie Tauber, who also published a very nice publication, and we will then have a common discussion, as you all are aware of this kind of format. So without further ado, please, Anna, we are very much looking forward to your presentation.
So thank you very much for this kind introduction, and thank you for the opportunity to present in today's webinar. So as already mentioned by Mario, our work focused on human granulomatous inflammation, and we used sarcoidosis as a model disease to investigate granuloma formation and also granuloma persistence. So this project initially was done at the baseline time point from a clinical study where we investigated new treatment approaches for granulomatous inflammation, and it was a collaboration between 3 labs in Vienna. So first, this Stary lab, which is led by Georg Stary at the Department of Dermatology from the Medical University of Vienna, where I am also part of. Second, the lab from Christoph Bock, which is located at the Center of Molecular Medicine, and also Thomas Krausgrube and Daniele Barreca are part of, which are also both first authors from this publication. And finally, the lab from Thomas Weichert, located at the Institute of Genetics at the Medical University of Vienna. So our lab was primarily involved in the supervision of the clinical trial, and we mainly handled the human samples, so the also the processing. The lab from Christoph Bock was involved in generating the bioinformatic results, and finally the Weichert lab tested the results in a murine sarcoidosis model, and of course all of the 3 labs interpreted the results together. So as already mentioned, we used sarcoidosis as a model disease to investigate granulomas. So sarcoidosis is an inflammatory condition which is characterized by the formation of these typical granulomas, which then lead to persistent and chronic inflammation and eventually organ damage by tissue fibrosis. So this image here shows the clinical representation of one patient with plaque sarcoidosis in the face. And here next to it, I have depicted the lesional skin biopsy from this patient, where we stained via immunofluorescence for the main components of the granuloma, with macrophages colored in red, T cells colored in blue, and fibroblasts colored in green. And we can clearly identify these granuloma structures in the dermal compartment of the skin. The etiology of the disease still remains unknown, but it is thought that it is a mix of genetic predisposition, environmental trigger, and a dysregulated immune response, which then leads to the formation of these inflamed granulomas. And then further, the interplay between activated macrophages, fibroblasts, and T cells results in persistent inflammation and eventually organ damage. So to verify this hypothesis, we dissected lesional and non-lesional skin biopsies from 12 patients with histologically proven cutaneous sarcoidosis, and we used single-cell RNA sequencing and combined it with spatial transcriptomics and immunofluorescence protein staining. So this UMAP here shows our entire single-cell sequencing dataset from lesional and non-lesional skin, and the lesional skin cells are colored in red and the non-lesional skin cells are colored in blue. We then further identified 20 different clusters, which included different T-cell clusters, myeloid cells, and structural cells. And what was particularly striking was that in the subpopulation of helper T cells, we found lesional and non-lesional skin-specific clusters, and this was the same in the subset of macrophages and fibroblasts. So we then further wanted to locate these different cell clusters in lesional skin. And for this, we used spatial transcriptomics, and this enabled us to locate previously identified cell clusters, which we identified with single-cell sequencing in a skin biopsy. So this image here shows you the H&E staining from one lesional skin biopsy, and we can clearly identify the granulomas here as these dense and compact cell aggregates. This representation here shows now a spatial transcriptomic plot from a matched skin section where we first located lesional and non-lesional skin gene signature. And we can see that the lesional skin gene signature is mainly located in the area of the granulomas. And what was interesting is that even in lesional skin, we can find non-lesional skin gene signature in between these granulomas. So this shows us that these intergranulomatous areas are more of a healthy skin condition, and the inflammatory process is located to the granuloma area. So we further located the lesional and non-lesional clusters of macrophages, helper T cells, and fibroblasts, and we could always locate the lesional fraction to the granuloma and the non-lesional fraction in between the granulomas. So therefore, we will further refer to the non-lesional cluster as homeostatic cells and the lesional cluster as granuloma associated. So in our publication, we then further always compared the lesional cluster of macrophage helper T cells and fibroblasts to their homeostatic counterparts. But due to time restrictions, I will focus today on the macrophages. In the macrophages, we could see two different clusters, the homeostatic one and the granuloma associated one. We then compared these two clusters and looked at the differentially expressed genes, and we found mainly genes involved in the attraction of monocytes and T cells, in Th17 and Th17.1 cell signaling, and also in extracellular matrix remodeling. And I will now go further into the process of extracellular matrix remodeling. And for this, I have prepared some stainings for different for extracellular matrix components, and also for the fibroblasts, which are also known to be involved in this process. And to locate the granuloma-associated fibroblasts, we used the fibroblast activating protein Fab as a marker gene, as it was higher expressed in the GA fraction. We also stained for collagen 4, which is known to be present in the basal lamina, which separates the epidermal from the dermal compartment, and which is also known to provide a scaffold and stability. This image here shows collagen 4 in yellow in healthy conditions, and we can see it here in the basal lamina and also around vessels. So if we now look at inflamed conditions, we can see that the Fab-positive fibroblasts are mainly located here in the surroundings of the granulomas. And if we now put on top the collagen 4 staining in yellow, we can see that here the granuloma is significantly enriched in collagen 4, and we can see a lot of small vessels surrounding the granuloma. So if we now go back to the macrophages, we additionally saw a lot of metabolic pathways upregulated when we performed a gene set enrichment analysis. And this includes oxidative phosphorylation, glycolysis, and gluconeogenesis, and also fatty acid metabolism. And additionally, we also saw mTORC1 signaling, which is known to activate lipid biosynthesis through the activation of the transcription factor SREBF1, which we found inside the granulomas. And this activation of lipid and sterile biosynthesis activates the macrophages and helps them to grow inside the granuloma. So this visual representation here is a summary of what we found in our publication. Even though I did not go into detail in every process, we highlighted the main pathomechanisms we found, and these are extracellular matrix remodeling, angiogenesis and cell metabolism, and also the secretion of cytokine and chemokines in order to retain and activate immune and non-immune cells. So now that we have seen all these mechanisms, I will shift to the therapeutic application in the disease. And for this, we decided to target one of these main pathomechanisms, and we first targeted extracellular matrix remodeling. For this, we used an inhibitor for MMP12. MMP12 is a matrix metalloproteinase, which is known to cleave ECM, extracellular matrix components, for example, fibronectin and also collagen 4, and is involved in macrophage trafficking. And for this, we used a previously established murine sarcoidosis model, which was established by the lab from Thomas Weichert, where the knockout of TSC2 results in the activation of mTOR and in the formation of sarcoidosis-like granulomas in different organs. The mice were treated with this MMP12 inhibitor over 4 weeks, and we could see that there was a significant decrease in paw and tail swelling in the treated mice compared to the control treatment, which was a sign of decreased skin inflammation. And finally, I would like to present you also the results from the clinical study I mentioned earlier in my presentation, and this is also beyond this publication. So based on the findings of Thomas Weichert, but also based on what we saw in our publication, we performed a clinical trial at the Department of Dermatology from the Medical University of Vienna, where we treated 16 patients with sirolimus, an established mTOR inhibitor. And what we could see is that after 4 months of systemic treatment, there was a significant decrease in disease severity in the skin. And to further visualize this, I have 2 immunofluorescence stainings. So this staining here shows a lesional skin biopsy before systemic treatment, where we can see that the dermal compartment is filled with granulomas. And in the subsequent biopsy after the treatment, we can see that no granulomas are evident anymore. There are just some relics of giant cells, which we can see here. And for everyone who's interested, these findings will be published online in Lancet Rheumatology from next week, Wednesday on. So this already brings me to an end of my presentation. And as a summary, I can tell that we identified a network of interacting granuloma-associated macrophages, fibroblasts, and helper T cells, which promote inflammation and also persistence of the granuloma. They remodel the extracellular matrix, promote angiogenesis, and attract and keep immune and non-immune cells. And the targeting of extracellular matrix remodeling and immune cell activation through inhibition of MMP12 or mTOR shows a decrease in disease activity in vivo. And finally, I would like to thank all the people involved in this project and also you for your attention, and I'm happy to take your questions.
Thank you very much, Anna, for this excellent presentation. And we immediately move to Marie Tauber presentation, and the question will come later on. So just a short introduction of Mary. So Mary is a MD PhD. She did her residency in Paris. Then she did her PhD in Nicolas Gaudencio's lab in Toulouse, and now she has an associate professorship in allergology and clinical immunology at the Department of Lyon Sud in France. So please, Marie, the stage is yours for your presentation.
So hello, everybody. It's my pleasure to be here, and thank you very much for the invitation. So these are the 3 main people involved in this study. That was my PhD project. So we are going to speak about mast cell biology. As you may know, mast cells are filled with cytoplasmic granules, as you can see here, and their most famous receptor is Fc epsilon R1, which can bind and be activated by IgE and allergen. but more recently, another important receptor has been described. It's MRGPRB2 in mice and MRGPRX2 in humans. It's a key receptor regulating mast cell biology, and the activation of both receptors lead to mast cell activation and degranulation. And historically, mast cells have been divided into two main categories. The first one is the connective tissue type mast cells that have heparin-enriched granules, which allow their staining using avidin, as you can see here in the skin. They are located mainly in the peritoneal cavity and the skin. And the other category is mucosal tissue type mast cells that are found especially in the lungs and the gut. And we cannot use avidin to label it, but we can use a specific chymase, which is MCPT1. But many questions remained regarding mast cells, specifically regarding their development, their heterogeneity, and their tissue role. Sorry. So what we first did was to sort a control population of mast cell from the peritoneal cavity in mice using CD45 and KIT, and immune cells from the skin and the gut mucosa. We performed 10x single-cell RNA sequencing, and as you can see here, we found an almost unique population of mast cell in the peritoneal cavity, two very small population of mast cells in the skin, and one small population in the gut mucosa. We aggregate all these population in a single UMAP, and you can see that mast cells from the peritoneal cavity and the skin aggregates together, whereas the mucosal mast cells, so these one are the CTMCs, whereas the mucosal mast cells in the gut mucosa segregate far away. And we look for differentially expressed genes between both populations, and we found two very distinct transcriptomic signatures with the expression of some specific proteases in mucosal mast cells, such as MCPT1 and 2, the expression of CD31 or CX3CR1, which, as you know, is mainly a marker of macrophages. And conversely, in CTMCs, we found the expression of other proteases such as the chymase 1 or the CPA3, and some neuroreceptors such as the receptor MRGPRB2 or MRGPRB1. And what was interesting was that the expression of these two genes, MRGPRB2 and MCPT1, was mutually exclusive in the two populations, meaning that CTMCs were only expressing MRGPRB2 and mucosal mast cells only expressing MCPT1. So we wanted to use those mutually exclusive markers to follow both population of mast cell in the tissues, and we label it. And you can see here that in the skin we found only MRGPRB2 positive mast cells, whereas in the different gut segments we found the coexistence of both populations with very rare MRGPRB2 mast cells systematically located in some anatomical niches here in the muscularis, whereas the mucosal mast cells were very numerous and always found in the mucosa. Then we wanted to integrate our data set with publicly available data sets. For example, here the mouse cell atlas, and you have here the distribution of all tissue cells across organs. We selected the mast cell population using those two genes, and you have here the different mast cells. And we found that mast cells expressing MRGPRB2 were indeed found in the skin and the peritoneal cavity, but also in the uterus, in the mammary gland, and in the heart. And MCPT1 mast cells were only found in the gut mucosa and the small intestine. Again, in the different tissues, we found a conserved transcriptomic core between both populations of mast cell. Then we wondered about the origin of mouse mast cells, and to answer this question, we first reanalyzed our data sets regarding whether the mast cells were sorted from neonatal pups or adult mice, and we found that MRGPRB2 positive mast cells were found both in neonatal pups and adult mice, whereas MCPT1 positive mast cells were only found in adult mice, meaning that the first population is from embryonic origin, whereas the second population developed after birth. And in line with this hypothesis, we stained both populations in a fetus E18, and we found only MRGPRB2 positive mast cells. So we wondered whether this population, which is located in the gut mucosa, so close to the microbiota, could be influenced by microbiota colonization. So we used germ-free mice, and again, we stained both populations, and we found a strong decrease in the number of mucosal mast cells in germ-free mice compared to wild type. Whereas the number of MRGPRB2 positive mast cells was basically unchanged, and here we are in the scheme. Then we settle a mast cell depletion mouse model based on the expression of MRGPRB2, and you have here the effect of the mast cell depletion in the peritoneal cavity 8 days after the first diphtheria toxin injection, and you can see that there is a slow replenishment that began at day 90. Of course, we checked whether the other circulating immune cells were affected by the depletion, especially basophils, and it was not the case. Then we applied to this mast cell depletion model two models of passive systemic anaphylaxis, two models of anaphylactic shock. Sorry, the first one is the passive systemic anaphylaxis model, and the second one, the active peanut-induced food allergy model. And you can see that in mice lacking the MRGPRB2 positive mast cells, they are almost protected from the development of anaphylactic shock. Here, that is measured in mice by the drop in body temperature, meaning that the mucosal mast cells that are still in these mice have a really dispensable role in anaphylaxis. Now, to finish, what about human mast cells? Well, we took advantage of a publicly available data set. Here it's the Tabula Sapiens. We selected, again, mast cells, as you can see here on this UMAP. And surprisingly, there was no obvious CTMC-MMC transcriptomic dichotomy in humans compared to what we found in mouse. So we performed an unsupervised nearest neighbor analysis. And as you can see here on this cluster dendrogram, we identified at least 6 distinct mast cell subsets in humans. We also determined a specific core gene signature in those 6 mast cell subsets, as you can see here, but I have no time to go into detail. And we look for the distribution of these 6 mast cell subsets across organs in human, and we found that they were well distributed in all organs, but some clusters have some specific tissue localization, for example, with mast cell cluster 5 in the lymph node. So to conclude, there's much complexity, transcriptomic heterogeneity of human mast cells that goes far beyond the classical CTMC-MMC dichotomy that is observed in mice. And there are many questions that remain. For example, could mouse and human mast cells fulfill a microenvironment specific functions such as tissue defense or support? What about mast cells under inflammatory conditions? Because as you saw, here we are at steady state. And could we specifically target some mast cells population depending on the affected organ? So I thank all the Inception Lab and collaborators, and I thank you very much for your attention.
So thank you very much, Marie. This was an impressive presentation. I think also, Anna, what you did was really, really nice and perfect examples showing first how rich the data sources need to be in order to publish high impact. But secondly, also how these novel methods place into the forefront previously under-researched cell types like mast cells, fibroblasts, and also macrophages. Thanks, you too. Thanks a lot. Please, you can always feel free to ask your questions in the chat from the audience. Meanwhile, if you are okay, Sandrine, I would start with the first question to Anna, and that is something that I always wonder. If you look in the immunology textbooks, especially the older ones, there's always written sarcoidosis is a kind of an anergic T-cell disease. It's a kind of immunosuppressive disease. So there's more inhibitory signals than activating signals. Now, I think you clearly show that this is not really true. And we are in the era of checkpoint inhibitors, where we do see that activating the immune system using checkpoint inhibitors frequently actually results in sarcoidosis. So would you say from your perspective and your data that sarcoidosis is an exaggerated immune response, or is it overwhelming T regulatory response or anergic T cell response? Can you bring light into this riddle?
Thank you for this very complex but very interesting question. Yeah, there are a lot of hypotheses where it comes from and what triggers these granuloma formation. Actually, we also saw both pathways. So we saw on the one hand this activation of immune and non-immune cells through interferon gamma secretion from Th17.1 cells and GM-CSF secretion. But we also, for example, saw the expression of checkpoint inhibitors for like CTLA4, PD-1, PD-L1. So there is also this exhausted T cell phenotype, which is also present. So I guess it is some kind of mixture of both. That is also why these granulomas persist in the tissue. But yeah, there are a lot of different hypotheses. There is also the auto-antibody hypothesis against vimentin. There are different environmental triggers which are thought to induce sarcoidosis. Like there is this fireman story who had a higher susceptibility to have sarcoidosis, and it's thought that it's because of inhaled antigens. So I cannot clearly respond to the question.
Okay, thank you. Maybe I can ask another question to Marie. Marie, you show very nicely that in germ-free mice, you have this strong reduction of mucosal mast cells. Do you think that these mast cells exert kind of non-allergic function in the body, that they are more linked to innate immunity than to allergy?
Yes, I think so, because as you saw in the, well, it was in mice, of course, but in the model of food allergy, we show that they play a minimal role in the development of anaphylactic shock. And it was a bit surprising because we found these mast cells very close to the food allergen, actually. So we could think that their role was important in food allergy, but it was not the case. So yeah, I think it's more a kind of macrophage role, like of surveillance or tissue homeostasis that they play. And I think we should develop a model of mucosal mast cell deficiency in order to more specifically study their role.
Okay, yeah, so a lot of work to come then.
Yes.
Can I briefly
Yes.
ask, Mary, was it technically difficult to have single cell sequencing from mast cells? Because usually
Yes,
with people,
very
they just throw it
difficult.
out. Any
Yes, because
trick you want to share, or is it your
it
secret
was.
and you have to collaborate?
No, indeed, it was very difficult because mast cells are very stuck in the tissue. You know, it's very difficult to sort them. So I think we just digest and digest and do many experiments until we found some of them. In the skin, there is a lot, so it's quite easy, and in the peritoneal cavity in mice, it's very easy. But it was much more difficult in the gut, and but I think that even if you have a small population of mast cell, you can study it.
Thank you very much for this excellent presentation and for answering the question. There are no additional questions into the chat, so maybe we can move on to the next slide. Thank you. So it's my pleasure to introduce the next ESGR Kitchen that will take place on the 14th of February at 1:30 as usual. It will be a talk given by Diamond Tassi and Louis Puig about the journey from PSO to PSA and can we prevent it. Yes, so all these episodes are, of course, available on YouTube, where you can not only watch this one, but also some previous one. I would like to advertise you about the next ESGR meeting that will take place in Lisbon, in Portugal, from the 4th or 7th of September 2024. And we are really much looking for all of you to this meeting. Very important dates. You can submit your abstract already from the 1st March on until the 3rd of June. And for late breaking abstract submission, it will be in July from the 1st until the 19th of July. And registration are opening in 2024. So now it's my pleasure to close this episode, and we are looking forward to see you in the next one. Thank you to the presenter and thank you to my co-chair. And again, thank you for our sponsor.